Welcome. This video will be a quick introduction for new users of OMERO at the University of Arizona. Before we get started, this slide's a reminder that OMERO is only intended for the storage and management of de-identified data. Now, we do have some groups doing patient-related research who have image data that incorporates a unique identifier within the file name. The unique ID relates the image data to a similar ID within the REDCap software, which is for storing clinical data, or the Open Specimen Biorepository software that's designed, both of which are designed for this type of data. The point being that it's not impossible to work with patient-related data, but you do need to be careful because protected health information of any kind cannot be stored in OMERO. This includes information embedded in the bitmapped image format, in other words, in the pixels of the image. And that would include things like medical imaging data or slide label images, as well as in the image metadata when the image was acquired or perhaps added by the user later or using tags, key value pairs, or other annotations that are available in OMERO. Well, we have a fairly standard OMERO installation, there are parts of our network configuration that are somewhat specific to our university. That's primarily because we use a virtual private network, or VPN, to control access to the server. People who don't have a U of A net ID will not be able to see or access our servers. If your lab has off-campus or non-U of A users who are willing to become what are called designated campus colleagues, the DCC process allows them to obtain a U of A net ID. This will enable them to access the UA instances of OMERO through the VPN. And there's a link in the notes that accompany this video for how to find out more about that process. And if you're simply interested in trying out the open microscopy environment and want to access a demonstration server that's actually not part of our institution, the open microscopy environment gives people anywhere in the world an opportunity to set up an account on their demo server. And there's a link in this slide and also in the notes for this video. We've had OMERO running here at the U of A on hardware located in the data center since the fall of 2022. We have two servers, a production server, which is primarily intended for research projects, and that's to help those projects comply with the data management requirements from the NSF and the NIH. Has about 100 terabytes of allocated space. There are a few projects on it, and we'd be happy to take more, and you can use the email address on this slide and in the notes below to contact us for more information. And in addition, we have a smaller training server that's primarily intended for preliminary testing and people that want to try out specific data imports. OMERO uses the Bioformats plugin, which you might be familiar with from working with ImageJ and other software that takes advantage of that. And it's able to open over 160 different proprietary file formats. But we do acknowledge that there are some formats that import better than others. The training server is also useful for new members who want to practice with OMERO before they start working with the production server. OMERO can accept data from nearly any instrument that creates digital image data. The image data can be multidimensional, X, Y, Z, time, wavelength. And while OMERO is primarily geared towards microscope image data, it can also be used for gel images, DCOM data, such as used in medical imaging, as well as other types of bitmapped image data. OMERO handles large image data like multi-gigabyte microscope slide scans very nicely. And the data can be uploaded to the server using a desktop client or a Python script. And we'll talk considerably more about the desktop client later in this video. The desktop client is not required to view or work with data that's already on the server. You can easily view OMERO data from a web browser without the need for additional software. Each lab's data is stored in a protected group with permissions about what can be done with the data and who can see it. and a companion video will cover managing and working with image data in groups. Currently, OMERO is free to University of Arizona users. The server and technical support are funded by the University of Arizona Health Sciences Strategic Plan 5.3. There are a number of other projects funded by the 5.3 initiative, and we encourage you to follow this link to learn more about Soteria, XNAT, and some of the other aspects to 5.3, and this link is also in the notes below. In August of 2023, we held an on-site workshop with the OMERO training team, and all the recordings from the workshop have been edited and are available on the D2L learning software. Interested users from the University of Arizona are welcome to reach out to us using the email that's provided to be given access to the videos. In addition, there are a number of resources that are on this web page on the U of A Microscopy Alliance that I regularly update. A list of the D2L videos is available from the Alliance webpage and also in the notes below. And please note as you look around online that there are a wealth of excellent YouTube videos, often done by the same trainers who did our workshop, but I would caution that you only watch videos that are no more than maybe around 2 years old, as some software tools in OMERO are regularly improved, they're added, or sometimes they're depreciated. If you want to work with the OMERO servers at the University of Arizona, you're required to use the university's virtual private network, even if you're physically sitting in a campus building that is already on the U of A network. To access the VPN, you need to have installed the Cisco AnyConnect software and have it configured correctly. There's a knowledge article, and there should be a link in the notes below, that the University Information and Technology Services has provided, and it walks you through how to get that software installed on your computer. And please note that if your computer is managed by one of the campus IT groups, you may have to have them install the VPN and also the OMERO.insight desktop software for you. If you don't have the VPN set up and running on your computer, and you try to connect to one of our OMERO servers using a web browser, you'll find that Chrome gives you a message like this, Firefox, and Edge. They can't see the server at all. With the Cisco AnyConnect software installed and configured, we recommend that you use the vpn.arizona.edu/full. For some reason that seems to work a little better than the standard campus VPN. To access the VPN, you log in with your U of A NetID. You'll find that it asks for your two-factor authentication, which I just approved. And once the VPN connects, if we go back to the web browser now, we can see that we have the login screen for OMERO. In OMERO, we currently use our UA NetID names to log in, but not our U of A NetID passwords. This is because OMERO doesn't work with Shibboleth, the university's single sign-on method, quite yet. They're working on it. For now, we set up new users to use their UA NetID name, but we give them a different password for accessing OMERO. And you'll need to be configured in advance as an OMERO user before you can actually log in. So make sure to contact us at the email address that we provide below to request access. Now that we're here, you notice that down at the bottom of this window, you can download the OMERO Insight client for the Mac OS, Windows, and Linux from here. Alternatively, you can search Google for OMERO downloads, and if you follow the first link that shows up, you can find the OMERO clients or OMERO Insight for Windows, for Mac, and for Linux. In addition, you'll find the plugins, for example, for Fiji and MATLAB if you want to work with them directly or if you're using IC. The Web addresses or the URLs for our servers, once you're in the VPN, are omero.arizona.edu, or if you're working with the training server, it's omero-training.arizona.edu. And OMERO will run in any modern web browser. Most of the development efforts of the open microscopy environment have been towards enhancing the OMERO web because it's not as dependent on a particular operating system. There is one other way to connect to OMERO, and that's through the command line interface called OMERO CLI for command line interface. If you're someone who understands Python, you can perform many useful functions through the use of scripting. And scripting can be handy if your lab will be routinely uploading image data to the server. Now, we may be able to connect labs with people with the skills to help them set up specific scripts. I am not a Python programmer, so I don't have much that I can say about Python or the command line to access OMERO. So let's log into OMERO Web. You may be wondering, why would I use OMERO Insight versus OMERO Web to connect to the OMERO server? The primary advantage and the purpose of the desktop client is for uploading data. If you're going to be working with and visualizing data that's already uploaded, then you should use the web client as it has more tools for visualizing and annotating the data. Before we go too much further, let's go over configuring the OMERO Insight desktop client. In the OMERO login window, if you click on this wrench right here, you're taken to a settings window. And you can add the domain names for our servers. If you just click on this little plus sign down here, and you can individually at least input at least one of the domain names that are shown here. And if you have access to multiple OMERO servers, perhaps at another university, you'll need to use that same tool to add those domains to this list so that you can access them when you need to. And you'll need to make sure that you select the correct server. If you don't pay attention, you'll find yourself trying to log into the wrong server with credentials that probably don't work for that server. So in this case, I selected the production server and I'm going to log in. So here I've logged into the production server, and on the left you can see OMERO Insight, and on the right you can see the OMERO web client. And something I learned just as I was making this video, periodically you may need to update your OMERO Insight. I wasn't able to log in with my 2-year-old version this morning because when the server gets updated, sometimes the Insight client gets out of date. Just tuck that away as if you have trouble logging in with Insight, it's possible that the client you have is a little too old for the version of the server we have. In OMERO Insight, you can see that I'm in my default group, which in this case is called Test Group. And if I click Display Groups on the upper left, you can see that there are a couple of other groups that I'm part of, or I could switch to a different group. In OMERO Web, if I put the mouse in the place where it says my name right here, I can actually show more groups. Now, what's interesting is I shouldn't be able to be part of these groups, but it shows all the groups that are on the server. And I'm not logged in as an admin. So this would be how I would switch to different groups in OMERO Web. In general, depending on which tool, desktop or web that you're using, you'll only see the contents of one user group at a time, particularly in the web browser. As an aside, in OMERO Insight, if I want to change the information about myself on the server, I can go up here in the group, click on my name, and I see this information about myself as a user. I have my login name, my first name, last name, my email address, what institution I'm from, and I can even upload a photo of myself if I want to, or I can change the password. And that might be because the one that you were initially assigned is not one that you'd prefer to use. We do request that it be a good password, keeping in mind that we don't have password rules like the U of A does. If you look at OMERO Web, you can see that in the upper right-hand corner where my name is, I can drop down this menu and choose User Settings. And I have username, first name, last name, email, institution. And you can also choose a default group, and that's actually true in OMERO Insight as well. And this is the group that shows up when you first log in. Now, just and a side note, everyone should belong to what's called the common group, which is mostly used as a holding area in OMERO for the purposes of the fact that if in a group that the group owner needs to delete you for their own personal reasons, you need to belong to at least one group or else they can't delete you. Or if they tried to delete you and you didn't belong to one group, all the data that you owned would potentially be deleted. So it's good to have a place that just everyone belongs to so that if you need to remove someone from a group without losing data, that's why we all belong to the common group. You can see that it's right here. Anytime you make changes to your profile, such as your password or updating your photo, please remember to click Save so that those are actually retained. So let's take a quick look at how data can be organized. We'll talk more about this in a later video. In OMERO, we have groups, and it may make sense for a faculty member to have more than one group for different types of data, with the caveat that sharing data between groups is kind of tricky and can have some challenges. And sometimes the sharing of data can be more easily resolved just simply by deciding who's allowed to be part of the group or groups that you're working with. Within a group, the top level of data is organized by projects. So that's what these purple folders in OMERO Insight are. And the dark blue folders over here in OMERO Web are the same thing. If I open a particular project folder on both sides, you can see now that we have a data set, and there can be several data sets within a project. And if I open this data set, you can see there are three unique pieces of data in that data set. There can be more data than that within any given data set. If I click on this particular piece of data in both OMERO Insight and OMERO Web, you can see that there's more information in the metadata window here or here. You can see that this file was acquired a little more than 10 years ago now. It's a .lsm file, which is an old Zeiss format. And you'll see in this window here how big the file is in pixels. And we have options to give it a tag. So if I drop this down, you can see it actually has one tag already. And I've tagged it as an LSM file, and I'll show you that over here as well. In OMERO, tags can be part of tag sets. So if you wanted it, for example, to have a tag set that listed the types of microscopes that you used, you might use a Zeiss confocal, Zeiss LSM 880, or a Nikon AXR, or an Olympus 4000, something like that. You could put that in there as a tag set that became an equipment tag set. And then you chose the appropriate one from that. There's just two levels, tag sets and the nested tags underneath it without options for any deeper levels. But you can create a large number of them based on experience from learning from other people, I have developed the opinion that every tag should have a description so that lab members are really clear about what they should be using to tag things. If there's any uncertainty, if things don't properly get tagged, then you'll have a harder time finding them. We can also have key value pairs. We don't actually have any for this one, but key value pairs may be as simple as tissue equals brain or liver, species equals mouse, or maybe Balb-C mouse or Sprague-Dawley rat or some sort of transgenic animal that you're working with. There may be information related to other data that we can't include directly in OMERO, like a REDCap database record number, which could securely connect this data to the protected information about clinically related samples that keeps us in compliance with HIPAA. In addition, there's the possibility to attach tabular data. Again, we don't have any on this particular one. And you can sometimes create that when you're doing image analysis. A number of the open source image analysis tools can access the data directly from OMERO. And then once you've done your analysis, you can upload the tabular data from your analysis back into OMERO. And you can put comments in, perhaps a pathologist's comments if you're looking at something with one of them. And I like this particular one under ratings. If you think this is the prettiest image you've ever seen in a particular experiment or one that you might want to consider when you're getting ready to publish at a later time, give it 5 stars or 4 stars, and then later on you'll be able to find it by searching for highly rated images. Let's look at the metadata that came with the images. If we look at the acquisition tab in OMERO Insight, you can see the original metadata here captured by this high-end microscope. And over in OMERO Web, if we go to the acquisition tab and we look at the original metadata, it's all the same data. And there's a lot of information in here. Some of it will be useful and some of it maybe wasn't or won't be. If we look here through here, we can see, for example, the objective that was used to capture this image. It's buried in there, but that information is useful. We can see the numerical aperture of the objective. There were a lot of different filters in here, and that same information is on both sides. So if we go over here to recording objective, we can see the same information about the lens. And that's extracted directly from the LSM file. Even though it's an older format, it's still imported fine. We should mention that there is no current standard for how instrument metadata is named or ordered. So that means that every vendor collects and names it differently. And this is currently an active area of discussion in the global microscopy community with the goal of creating a standard that meets the need of both the vendors and the users. And we'll be talking more about tagging, key value pairs, and metadata in a later video. If you want to change the file's name to something different from the name that it was uploaded as, you can go in, click on the pencil here, and you can change the name and then choose to save it. Same thing over here, click on the pencil, you can change the name. I would note that it might be harder to relate the OMERO data to the source file that you hopefully have backed up elsewhere if you decide to change the name after you have uploaded it. So that's existing data. There's also options for restructuring your data as well. Perhaps you want to create a new project. In OMERO Insight, we can create a new project. And we'll just call this practice. We now have an empty project. If I take this last image and I decide I'm going to put it in this project, you notice that nothing happened. That's because it wants me to make a data set first before I move it over there. So let's do that. We'll create a new data set, call it test. Not particularly clever with names, but I'm not going to keep this. So I have the project, the data set, and right now the data set is empty. If I take this one image and drag it up to the data set, takes a moment, but then it updates it. If I want to see those changes reflected over here in the web client, if I hit refresh, you can see we now have a practice folder with test and that one file. I don't want to leave this way, so I'm actually going to move this file back. And I'm going to delete this group. So I don't want to have that there. And over here in Insight, if I click these two arrows, that will refresh that, so that updates as well. And we'll talk a little bit more about working with data, but a couple of key points. If I wanted to delete that image, I could, since I am the owner. But that's only an option if you are the owner of the group or if you happen to be the person that uploaded the image data using your login. We'll talk more about data ownership, group permissions, sharing, and managing a group in the next video. Now, sometimes people ask me, why doesn't OMERO have a much deeper file system? And that's a fair question. And we'll talk a little bit more about that in the third video in this series. But if you consider how Gmail organizes emails, it's all based on tagging. There's no actual folders in Gmail. And OMERO uses that same object-oriented methodology as opposed to using deeply nested folders. Now, if you want to look at how things are tagged, we can go over here to this tab that says Tags in OMERO Web. And you'll see that we have data that was tagged with the Leica SP5 confocal, the LSM tag. We have things from the Axio Observer, which is down the hall from my office, and things that were specifically tagged with the more modern LSM 880. There's also a Western blot. So you can see that the tags allow us to subset the things within the larger list of data. I could have tagged things as slide scans, for example. If you want to add a new tag, let's go back. So let's go to some slide scans. And I'll pick this last one. Click on tags, and you see the plus sign here. And I'm going to create a new tag and give it a name called slide scan. So I've given it a name, I've added a description. Hopefully yours isn't as lame as mine, but that works. Now we've got slide scan, we save it, and it's tagged that way. If we go back to the tags list, we should see that as an option. You can also do tagging on a data set level. Everything within this data set is a slide scan. We can tag the data set by doing slide scan, save, and now if we want to go back to tags, we see that there is both a data set and an individual piece of data. But if we wanted to do them all within that data set, we should be able to just tell it that all of them are slide scans, save. Now, if we look at an individual image, you can see. So all I did was a shift click where I did the top first and the last with holding down the shift button. And now we have that in all the individual images within this data set. In addition to the data set, are labeled as slide scans. And you can imagine if we did things with key value pairs that would be useful. And there's actually a tool for searching that called OMERO Parade, but we don't have any key value pairs right now. Let's just look at OMERO Parade briefly. So you can be looking through a data set and ask it to filter it based on regions of interest, its rating. So let's see if we can find the 5-star images. Oh, we lost one because only 2 of them are rated as 5 stars. So we could add a different filter instead of our ranking. We could look for tags. And again, this is only going to show us the tags that are available within this data set. If I go to the group level, you notice I don't have an option. So there's still some things about Parade I'm learning, but there are ways to subset your way down into complex searches. So an important thing to consider about OMERO is that we're really trying to avoid OMERO becoming a fancy version of a box folder or any other hard drive space, Dropbox, SharePoint through Google Drive. And unfortunately, we've seen examples where labs just throw data in there and someone figures out later, if they can, what it is and how it relates to the lab's research. We really want groups to use tags and key value pairs consistently so that when they look for specific data, they can actually find it relatively easy. And for tags in OMERO, using some tips from the German bioimaging group, use tags as a way for substituting for your deep folder hierarchies. You can think of it as a way to do file paths, and you want to have that be intuitive so that anyone can figure that out. And key value pairs would enrich your data in a biologically meaningful way about the sample, the preparation, the staining methods, the genetic background, antibodies used, all the stain used, all those things that talk about the experiment itself. Because those are not the things that are captured by the microscope, assuming that you have a highly automated microscope. And there is an effort internationally to come up with a list of things. We encourage you to look at the link that's provided below for the recommended metadata for biological images, sometimes called REMBI, and that gives you a lot of information. Consider how you structure your data and standardize your terminology. There's a number of well-vetted biological ontologies or structured vocabularies. And you can see the article below called 10 Simple Rules for Selecting a Biontology. Consistency is super important because, as many of you probably discovered when you're searching with computers, if half your lab is using the word renal to describe the images and the other half is using kidney, you're not going to find half the images if you use one of those words. You can use tags and key value pairs and structure your data that way. We want OMERO to be in support of the FAIR principles where data is easily findable, it's accessible, it's interoperable, and it's reusable. And since your lab is the most likely future user of this data, do yourself a favor and annotate it well. You know, the joke is that future you can try to reach out to past you, but past you does not respond to emails. So past you has to leave enough clues for future you to be able to reuse the data. If you want to add new data to a group, OMERO Web doesn't do that. You'll need to use OMERO Insight. You can upload image data using Python scripts, but I don't have any Python expertise to offer here. If you go to OMERO Insight and use the upload icon, this upward facing arrow here, you open a new dialog box. And from here, you can search for locations on your computer. The first time I recorded this video, I went to upload some data from our super resolution microscope, and it took forever. So I'm going to upload something that actually isn't part of a microscopy data set, just to show you that you can upload things that are not specific to microscopy or to biology. This actually happens to be a large image of a world map, and I had to convert it from JPEG to an OME TIFF, and I'll explain that. So I'm going to put it in a new project called map, and the data set is going to be called color map. So I'm going to be in the process of creating a new project and a new data set. I'm going to add it to the queue, tell it to import. And the reason I brought up the information about the ELIRA data is because that was several gigabytes worth of data to upload, and it took a long time because I'm on Wi-Fi. So I encourage you, if you're uploading data, particularly data of a good size, to be on a wired computer as opposed to wireless. It will go so much faster. Now, the reason I had to convert this from the JPEG to the OME TIFF is when images are above, particularly large images like this, are above a certain size, just like a slide scan, OMERO wants this to be in what's called a pyramidal file format. Much like Google Earth, if you're looking around Tucson and you want to see your neighborhood, you don't actually upload the entire city of Tucson in high resolution to be able to look at your neighborhood. The pyramidal file format takes a lot of strain off of the server, and unfortunately, it doesn't do the conversion itself, so you have to do that in advance. So I've uploaded it. I'm going to refresh. There's the map. Let's go over here and we're going to refresh the web browser. And you can see that we have map, color map. And if I double click on this image now, I go into what is called OMERO iViewer. And if I zoom in, you can see there's quite a bit of detail in this image. I can pan around. And down here you see the thumbnail of where we are within the world, if you will. So we want to go up here to where Arizona is, where there's no rivers except the Colorado. So just to show you the utility of this tool, we can close this window and let's go back to one of these old Zeiss files and double click on it. Now we can go up and down in the Z stack. It's a little hard to see, isn't it? So let's change the histogram a little bit. And now as we go up and down, you can see we have different data at different levels. You can see we can look at the multi-dimensional data here. If I wanted to change this color to red, it's that easy. Change this to cyan, and it looks totally differently. The beauty of this is the things that I have done here do not affect the raw data. They only affect the display that I see. Couple notes here. Bioformats is an amazing tool in OMERO, and you've probably used it in Fiji or ImageJ. It can import over 160 different proprietary file formats, but it's not perfect, and some formats are not supported or they're not very well supported. And that's because the software developers for bioformats are usually having to reverse engineer all of the details of how the file format works because the vendors often give them little or no help. And you can review the list of supported file formats. There's a link in the notes below. Large images don't import well because the server is not well configured to do that image processing to convert the file into that pyramidal file format. It will upload, but it may take an entire day for the server to convert a single large file to allow it to be viewed. And a folder's worth of images could take weeks. So the better solution is to go in and pre-convert the images to an OME TIFF file. For example, we've discovered that files from the Nikon slide scanner over in the Cancer Center need to be converted to the OME TIFF. There is free software that can be downloaded from Glencoe, which is the commercial arm of the open microscopy environment. It's called the NGFF converter, or Next Generation File Format Converter. And we have a workstation in Life Sciences North with the software on it. The great thing is you can do a batch conversion. So if you have a lot of files to convert, you tell it everything in this folder, change it from this format to the OME TIFF. I'll come back in an hour or two. And one of the reasons that we have a training server here at the University of Arizona is to allow labs to test and see if new file formats are going to work before they upload files to the production server. That way, if there's a problem with a new file format, we may be able to work out a way around it. And we do keep an eye on the public discussion forum for OMERO and also for other image analysis software located at forum.image.sc. There are several videos from our August 2023 workshop about the features of the image viewers built into OMERO, but I'll just give you a few highlights here. You can load the image viewers by dropping this down. I've already loaded a couple of images in the viewers, so let's just go through these tabs here really quickly. There is a very rudimentary viewer in OMERO Insight, but you really want to use OMERO Web to look at your image data. So this is the built-in viewer. It has some strengths. It's very easy to split the channels. You can do a maximum intensity projection of this Z-stack very quickly. And we can do some of the same things we did before as far as moving up and down in the stack. But you can see it's a little harder to adjust the channels to get more information out of them. So if we go to the OMERO iViewer, which is what we looked at before, if we tell it to do a minimum, maximum, and we bring this down. Why it turned that off, that's why it's not seeing it. So I'm going to bring this up so we can see it a little better. And then as we go to different levels, you can see different information. You can see it's actually saturating. So let's back it off a little bit. And as I showed you before, we can turn channels off. We can change the colors of channels to different things, and it's that easy. And again, the underlying data is not changed. Within the viewer, we can also put regions of interest. And we can give it names. And so other people can come in and see what it is that you thought was amazing. And I would encourage you, if you're going to annotate images this way, that you use a fairly standard nomenclature so that you can find things based on these regions of interest and their annotations. There's also ways where you pull the data out into other programs that maybe draw circles around your nuclei, and you can upload those regions of interest back into OMERO. You notice that this view has a scale bar. Again, we can go up and down and see different data. As you go to more complex data, here is data that was scanned spectrally by the LSM 880 confocal on main campus. And we have all the different spectral channels displayed here. If we go to the spectrally unmixed data now, we have a very large data set in X, Y, and Z with multiple colors, including the residual from the autofluorescence that was unmixed. Another nice viewer that's available is the 3D viewer. This is called FP Bioimage. It's actually based on some rendering from gaming. So you can see we can rotate this around in three-dimensional space. And rendering this was pretty quick. Now this is just a single area Z-stack. Something larger might take a little bit more time. So that gives you an idea of some of the things that you can do. And the final, more interesting viewer to a lot of people is OMERO Figure. An OMERO Figure allows you to build a composite image, much like you would do in Photoshop, without nearly as much pain. The beauty of Figure is that any changes made do not affect the underlying data, and you can share that figure with other people who may or may not want to change it a little bit. So let's go to OMERO Figure, but I don't want to do just that image. I want to do a new one, and we're going to do a composite. And I'm going to use these image IDs. You see here that there is an image ID number. So every image has a unique ID. You can see this one is a different number. So I have a list of them. I'm just going to type them in and ask it to add the images. Interested in these images, but I want to do some things with them. I want to label them with scale bars. I'm going to put 50 micron scale bars on these images in black. Look at that, it did it to all of them. That's excellent. But I'd like to look at this image a little more closely. So if I go to preview, I can zoom in. You notice that the scale bar now has changed to reflect the zoom in function that I did on the image. And if I look at this and I go, what I really want is the image that this image right here was zoomed from, I'm going to select this one, delete it, select this, edit, copy, paste. I'm going to select them all, tell it to align to a grid, and I'm going to come back to this one and zoom it back out. Oh, and zoom this one back in, sorry. So here's your original image, here's your zoomed in image. You could even put a region of interest on, wait a minute, I want it to be, I want to put a region of interest on this image so that I can tell people that's about the area that I zoomed in on. Not quite, but let's push that up a little bit. And there we go. So I did that all that fast. And while this isn't a fabulous figure, it gives you an idea of the potential that's here. There's a lot of interesting things that you can do with OMERO figure. So to wrap things up on this particular video, I want you to know that the next video in this series will go over group and user permissions and data ownership, and the third video will talk more about data management and metadata in more detail. So I wanted to thank you for watching this video. The next video in this series will cover group and user permissions as well as data ownership. And the third video in the series will cover data management and metadata in more detail. If you want to scan this QR code, that'll get you to the email that's here on screen if you need to ask questions. And again, just to be clear, the OMERO installation that we're talking about is for members of the University of Arizona. I'm sorry, we can't help everyone. Thank you. Have a good day.